Okay, so good evening, everyone. Today I'll be starting my live session now. Let me share my screen. Yeah. So I hope my screen is visible and I'm audible. So good evening, everyone. This is the 8th week live session of this course, Post Pathogen Interaction Immunology, taken by course instructor Professor Himanshu Kumar from Aishi Dasgupta. So what we basically do in this course live sessions is that we take some previous year old assignment questions and we discuss the nitty gritties of answering that question. We not only discuss the answer to that question, but we also discuss the surrounding concept so that it helps us solve similar type of other questions, right? My name is Aishi and I'm a PMRF research scholar from the Department of Chemistry, IIT Bombay, Maharashtra. And basically, we try to keep the sessions as interactive as possible. So if you know the answer, after repeating the question, I wait for some time. After ending the discussion, also I wait for some time. If you have any doubts, any queries, any other opinion, you can just unmute your mic or type in the chat box and let me know. If you have any queries, any doubts also, you can ask me immediately. If I know the answer at the moment, then I will say. If I don't, then you have to give me some time. I'll go back and give it a good reading, and then I'll be back with the answer by next week, right? So, with that introduction, let's start with the first question for today. Which of the following statements about lymph is true? Okay, I will repeat the question and then I'll wait for some time. I'll give you some time to answer the question, right? Which of the following statements about lymph is true? Option A. Lymph is formed only from tissue fluid containing oxygen. Option B. Lymph is rich in antigens that originate from tissues. Option C. Lymphatic capillaries are closed-ended structures with no openings. Option D. Lymph flow is enabled solely by the pumping of the heart. Option E. Lymph does not contain any immune cells. Which one do you think is true and why? Anybody? Okay. So the correct answer in this case will be uh, option B, which is lymph is rich in antigens that originate from tissues. Now, why? Let's analyze each option briefly. Option A is lymph is formed only from tissue fluid containing oxygen. Lymph is formed from interstitial tissue fluid, but it does not contain only oxygen. It contains many substances such as proteins, lipids, cellular debris, pathogens, antigens. So, this statement is incorrect. Option B, lymph is rich in antigens that originate from tissues. This is correct. So when tissue fluid enters lymphatic capillaries, it carries microbial antigens, cellular debris, foreign particles. So these antigens are transported to lymph nodes where immune cells like the lymphocytes and macrophages detect them and initiate immune responses. Then option C, lymphatic capillaries are closed-ended structures with no openings. So lymphatic capillaries are blind-ended, closed at one end, but they do have openings between endothelial cells that allow fluid and particles to enter. So saying no openings is wrong. Option D, which is lymph flow is enabled solely by the pumping of the heart, it's also incorrect because unlike blood circulation, lymph flow does not depend on the heart. It moves because of skeletal muscle contraction, breathing movements, smooth muscle contraction in lymphatic vessels, one-way valves preventing backflow. Option E, lymph does not contain any immune cells, also incorrect because lymph contains immune cells, especially lymphocytes like T cells and B cells, dendritic cells, macrophages, etc. So, the only correct answer among these set of options is option B. Lymph is rich in antigens that originate from tissues. So, we have talked a lot about the immune system, right? And how the body responds to disease and injury, about the non-specific response, which is the innate immunity, about the specific response, which is the acquired immunity, and all of these. Now, what do we mean by lymphocyte development? Basically, we'll talk about that. What do we mean by lymphocyte development? What's the origin, lineage, functions, etc. So, if you talk about that, all immune cells originate from hematopoietic stem cells or HSCs in the bone marrow. Now, these stem cells are multipotent, which means that they can give rise to all blood and immune cell types. From HSCs, two main progenitor cells derive, which is the common myeloid progenitor CMP and the common lymphoid progenitor. Okay. So as I said, rich supply of hematopoietic stem cells, asymmetric cell division, where like one daughter stays in the bone marrow. The lymphoid and the myeloid lineage cells begin and are released from here. Now the differentiation of the lymphoid stem cells in the bone marrow, the general B cells matured in the bone marrow, and the differentiation into lymphoid cells, stem cells in the thymus, general T cells matured in the thymus. Now, migration of mature general B and T cells to secondary lymphoid organs, basically the lymph nodes, the lymph node which is here, if you can see the spleen here, tonsils here, and external body surfaces, intestinal, respiratory, urinary, and reproductive. Okay, now if we talk more about this, so the myeloid lineage is derived from the common myeloid progenitor, and the cells produced include neutrophils, eosinophils, basophils, monocytes, which get converted to macrophages, dendritic cells, some types, mast cells, etc. The function is that these cells mainly participate in innate immunity, meaning rapid, non-specific defense against pathogens. The examples are neutrophils, phagocytosed bacteria, macrophages engulf pathogens and present antigens, and dendritic cells link innate and adaptive immunity through antigen presentation. Then we have the lymphoid lineage. It is derived from the common lymphoid progenitor. So cells produced include B lymphocytes of the B cells, T lymphocytes of the T cells, natural killer cells, and some dendritic cells. So these cells are responsible mainly for adaptive immunity, which is antigen specific and has memory. So B cells produce antibodies, T helper cells or CD4 plus coordinate immune responses, cytotoxic T cells CD8 plus kill infected or cancerous cells. Now NK cells or natural killer cells destroy virus infected cells without prior sensitization. Right? So the B cells development happens in bone marrow, maturation happens in bone marrow. T cells development happens in bone marrow, maturation in thymus. NK cells development happens in bone marrow, maturation in peripheral tissue. And myeloid cells development happens in bone marrow, maturation in bladder tissues. So the primary lymphoid organs involved are bone marrow, thymus. Secondary lymphoid organs where immune responses occur are lymph nodes, spleen, and the mucosa associated lymphoid tissue or mold. So the developmental system ensures that the immune system can recognize pathogens, destroy infected cells, produce antibodies, and develop immunological memory. So this coordinated origin and differentiation allow both innate and adaptive immunity to work together. Right? So immune activation, we know about the antigen antibody binding. What triggers these cells to respond? So the B cells recognize antigens, proliferate and produce specific antibodies. They can differentiate into plasma cells to produce more antibodies, differentiate into memory cells that keep antibodies in supply for activation from second encounter by the same antigen. So B cells recognize antigens, proliferate and produce specific antibodies. They differentiate into memory cells, keep some for later. So cell response, T cells recognize and destroy tagged antigens and proliferate. Cytotoxic T cells bind to antigen and plasma membrane of target cells and directly destroy the cells. Helper T cells activate B cells, cytotoxic T cells, natural killer cells and macrophages. So remaining cells can respond to secondary exposure. Now the cytotoxic T cell binds to antigen on plasma membrane of target cells and directly destroy the cells. Helper T cells activate B cells, cytotoxic T cells, natural killer cells and macrophages. So we'll not go into too much detail about this. We'll directly go about the lymphatic vessels. So they can carry lymph away from tissues. The lymphatic capillaries are more permeable than blood capillaries. Epithelium functions as series of one-way valves, right? So lymphatic system consists of lymphatic vessels, lymphatic tissue, lymphatic nodules, lymph nodes, tonsils, spleen, thymus, etc. Now, if you talk about the function of the lymphatic system, fluid balance, excess interstitial fluid enters lymphatic capillaries and becomes lymph. Fat absorption of fat and other substances from digestive tract, and obviously defense. Microorganisms and other foreign substances are filtered from lymph by lymph nodes and from blood by spleen. Now lymphatic capillaries join to form lymphatic vessels that have valves and ensure one-way flow. Lymph nodes distributed along vessels and filter lymph. Lymphatic trunks like jugular, subclavian, bronchomediastinal, intestinal, lumbar, and lymphatic ducts, right and thoracic, which connect to large veins. Okay. Now, lymphatic tissue consists mainly of lymphocytes. It can be encapsulated or not. And the lymphatic nodules basically numerous in loose connective tissue of digestive pairs, patches, respiratory, urinary, reproductive systems. Now, if we talk about tonsils, they are a large group of lymphatic nodules in nasopharynx and oral cavity. They provide protection against bacteria and other harmful material. The groups are palatine, pharyngeal, lingual. Okay, lymph nodes are organized in cortex and medulla. Substances removed by phagocytosis or stimulates lymphocytes or both. Only structures to filter lymph, afferent and efferent vessels. Now, spleen is located in the left superior side of abdomen. Can be ruptured in traumatic abdominal injuries resulting in bleeding, shock and death. Now, the blood flows through at 3 different rates: fast, slow, intermediate. The functions are destroys defective RBCs, detects and responds to foreign substances, limited reservoir for blood. The spleen. Okay. Now thymus, located in superior mediastinum, divisions, cortex and medulla, site of maturation of T cells. Immunity, ability to resist damage from foreign substances as microorganisms and harmful chemicals. The categories are innate or non-specific resistance, like mechanical mechanisms like prevents entry or remove microbes, chemical mediators like promote phagocytosis and inflammation, and cells like involved in phagocytosis and production of chemicals, all of that. And adaptive or specific immunity, like specificity, will be ability to recognize a particular substance. Memory, like ability to remember previous encounters with a particular substance and respond rapidly. Now, innate immunity, the cells will be white blood cells, most important cellular components of the immune system. The methods are chemotaxis, phagocytosis. Neutrophils, basically phagocytic and first cells to enter infected tissue. Macrophages will be monocytes that leave blood, enter tissues, large phagocytic cells. Basophils and mast cells promote inflammation. Eosinophils will be reduced inflammation. Natural killer cells, lyse, tumor and virus infected cells. If we talk about the inflammatory response, tissue injury, regardless of type, can cause inflammation. Response initiated by chemical mediators that produce vasodilation, chemotactic attraction, increased vascular permeability. The types can be local, like symptoms are redness, heat, swelling, pain, loss of function. Systemic can be symptoms are increased in neutrophil numbers, fever and shock. Okay. Either bacteria enter tissue damage occurs, chemical mediators are released, increased blood flow, chemotaxis, increased vascular permeability, increased number of white blood cells and chemical mediators at site of tissue damage. Then bacteria contain destroyed and phagocytosed, bacteria gone, tissue repair, bacteria remain, additional chemical mediators activated. Adaptive immunity basically involves the ability to recognize, respond to and remember a particular substance. Stimulants can be antigens like large molecules, foreign, not produced by body, introduced from outside. Self-antigens can be produced by body. Haptens are small molecules and capable of combining. The types will be humoral or antibody-mediated B cells, cell-mediated T cells. Now, with the original development of lymphocytes, B and T cells originate in red bone marrow, move to lymphatic tissue from processing sites and continually circulate. Clones are small groups of identical lymphocytes. Now, the positive selection ensures survival of lymphocytes that react against antigens. Negative selection eliminates lymphocytes that react against self-antigens. Now, primary lymphatic organs, red bone marrow, thymus, where lymphocytes mature into functional cells. Secondary lymphatic organs, where lymphocytes produce an immune response. What are the antigenic determinants? Antigenic determinants, specific regions of a given antigen recognized by a lymphocyte. Antigenic receptors will be surface of lymphocyte that combines with antigenic determinant. If you talk about the major histocompatibility complex or MHC, most lymphocyte activation involves glycoproteins of cell surfaces called MHC molecules. Now, the class 1 molecules display antigens on surface of nucleated cells resulting in destruction of cells. Class 2 molecules display antigens on surface of antigen presenting cells resulting in activation of immune cells. So, this is the steps of, So, what are the steps of antigen processing, if you ask? So, basically, these are the steps for antigen processing, right? So, basically, it refers to the cellular mechanism by which proteins or antigens are broken down and presented on the cell surface bound to MHC molecules so that T lymphocytes can recognize them. 1 minute. Right? So T lymphocytes can recognize them. So the steps differ slightly for MHC class 1 and MHC class 2 pathways, but the overall logic remains similar. Uptake, processing, MHC loading and presentation. So the antigen uptake, first the antigen must enter the antigen presenting cell. The cells involved are dendritic cells, macrophages, B cells. The mechanisms of entry will be phagocytosis, engulfing bacteria, large particles. Endocytosis, uptake of soluble antigens, and receptor-mediated endocytosis, specific antigen uptake by B cells. Then we have the antigen degradation or processing. The internalized antigen is broken into smaller peptide fragments. So this occurs in the endosomes, lysosomes for exogenous antigens, MHC2 pathway, and proteasome in cytoplasm for endogenous antigens, MHC1 pathway. So proteolytic enzymes digest proteins into peptides of suitable length. Then MHC molecule synthesis. So meanwhile MHC molecules are synthesized in the endoplasmic reticulum. MHC1 associates with beta 2 microglobulin in the ER. MHC2 associates with the invariant chain which prevents premature peptide binding. Then peptide loading into MHC. MHC class 1 pathway for intracellular antigens like viral proteins. Proteins degraded by proteasome, peptides transported into ER via TAP transporter. Peptides bind to MHC class 1 complex stabilized. These are recognized by the CD8 plus cytotoxic T cells. MHC class 2 pathway will be basically for extracellular antigens. The antigen is degraded in the endosome or the lysosome. MHC2 travels from ER to endosome, invariant chain removed. Clip replaced by antigen peptide with help of HLA-DM. Peptide binds MHC2 recognized by CD4 plus T cells, helper T cells. Now transport to the cell surface, the peptide MHC complex moves through the Golgi apparatus and is transported into the plasma membrane. Now the T cell receptor recognizes the peptide MHC complex. MHC1 will be CD8 plus cytotoxic T cells. MHC2 will be CD4 plus helper T cells. This recognition triggers T cell activation and immune responses. Okay, so this is it. Now if you talk about if you want to know about the co-stimulation, what is the first signal, what are the co-stimulation by surface molecules, then we have to talk about in T cell activation. The two signals are required. Signal 1, antigen recognition, and signal 2, which is co-stimulation, right? So without the second signal, the T cell becomes anergic or non-responsive. So the first signal or signal 1 is the antigen specific signal, right? So this signal provides specific recognition of the antigen. Components involve T cell receptor or the T cell peptide MHC complex on the antigen presenting cell. Interaction, CCR plus CD3 complex binds to the antigen peptide presented on MHC. Additional stabilizing molecule CD4 binds MHC class 2 helper T cell. CD8 binds to MHC class 1 cytotoxic T cell. So the first signal is TCR plus peptide MHC interaction. Now second signal is the co-stimulation by surface molecules. This signal ensures that the immune responses is activated only when necessary. So the major co-stimulatory interaction is CD28B7 interaction. Okay, it basically promotes T cell activation, induces IL-2 production, stimulates T cell proliferation. Now what are the additional co-stimulatory molecules? Other surface interactions that enhance activation include the T cell CD40L. CD40 activates APC and enhances immune response. ICOS-L supports T cell proliferation. ICAM, LFA1, ICAM1 strengthens cell adhesion. So what happens without co-stimulation? If signal 1 occurs without signal 2, T cells become allergic, immune response is suppressed, prevents autoimmunity. Okay. So this is the proliferation of the helper T cells. Now what do we talk about? What do we mean by lymphocyte inhibition? Right? So tolerance basically to prevent the immune system from responding to self-antigens, that is provoked by deletion of self-reactive lymphocytes, preventing activation of lymphocytes and activation of suppressor T cells. So lymphocyte inhibition refers to the suppression or downregulation of lymphocyte activation, proliferation or function in the immune system. Okay, it is an important regulatory mechanism that prevents excessive immune responses, tissue damage and autoimmunity. So the lymphocytes mainly include T lymphocytes, B lymphocytes and natural killer cells, right? So when these cells are inhibited, their ability to proliferate, produce cytokines or kill target cells is reduced. So what are the mechanisms of lymphocyte inhibition? Inhibitory surface receptors, immune checkpoints. Certain receptors on lymphocytes send negative regulatory signals. Examples are CTLA4, cytotoxic T lymphocyte associated protein 4. Present on activated T cells, competes with CD28 for binding to B7 on APCs, delivers an inhibitory signal reducing T cell activation. Then we have PD1 or programmed cell death protein 1 found on T cells, binds PDL1 or PDL2 on other cells, causes T cell exhaustion or inhibition. These mechanisms are called immune checkpoint pathways. Then we have the regulatory T cells. Regulatory T cells suppresses other lymphocytes. They inhibit immune responses by producing inhibitory cytokines, direct cell-cell contact. Key cytokines will be IL-10, TGF-beta. Then we have inhibitory cytokines. Certain cytokines reduce lymphocyte activity. Examples will be IL-10 suppresses macrophages and T cell activation. TGF-beta inhibits T cell proliferation and B cell function. Now, the lack of co-stimulation or energy for T cell receives signal 1, TCR-MHC interaction, without signal 2, which is CD28B7, the cell becomes anergic. Anergic lymphocytes do not proliferate, cannot respond to the antigen again. Then we have the activation-induced cell death. Overstimulated lymphocytes can undergo apoptosis through pathways such as FAS-L interaction. This eliminates excessive immune cells after infection. The biological importance: lymphocytes inhibition helps to maintain immune tolerance, prevent autoimmune diseases, limit chronic inflammation, terminate immune responses after pathogen clearance. Okay. Antibodies or immunoglobulins classes are IgG, IgM, IgA, IgE, IgD. The variable region part that combines with an antigenic determinant or of antigen, constant region response for activities. The actions of antibodies: they inactivate the antigen, binds antigens together, activates the complement cascade. The complement cascade activated, inflammation, chemotaxis, lysis, facilitates phagocytosis. Initiates the release of inflammatory chemicals and facilitates phagocytosis. So we'll not go into too much detail of this immunity. We already know about this. Now, what are the effects of aging? Little effect on lymphatic system, but decreased ability of helper T cells to proliferate in response to antigens, decreased primary and secondary antibody responses, and decreased ability of cell-mediated immunity to resist intercellular pathogens. So basically, the high hydrostatic pressure in the arterioles and capillaries at the arterial part of the circulation leads to move plasma fluid from the capillaries to the interstitial tissue. Some of this fluid returns to the bloodstream of the venous side, while the remainder returns to the circulation through vessels of a separate system that is called the lymphatic system. The fluid that enters the lymphatic system is called lymphatic vessels, is called lymph, and the lymphatic system is a part of the circulatory system. The main function of it is to return fluids escaped from capillaries to the circulation and also play an important role in defense and immunity. So tissue drainage, like every day around 21 liters of fluid of plasma carrying dissolved substances and some plasma protein escape from the arterial end of the capillaries and into the tissues. Most of this fluid is returned directly to the bloodstream via the capillary at its venous end. The excess about 3 to 4 liters of fluid is drained away by the lymphatic vessels. It returns the excess proteins to the circulation, remove waste products and toxins. Fat absorption is fat and fat-soluble materials. Fat-soluble vitamins are absorbed into the central lacteals, essential to the immune system. Components are lymph vessels, lymph nodes, lymph organs, and diffuse lymphoid tissue. Now, the lymph is a clear, watery fluid similar in composition to plasma with exception of plasma proteins. Lymph contains small amounts of proteins. Lymph contains cells mainly by lymphocytes, large molecules of fat like chylomicrons that are absorbed from the intestines into the lymph vessels. Now, blood circulation, lymphatic circulation and innervation of the small intestine. The smooth muscle system for contracting the villi is illustrated in the villus on the right. Now, the lymph vessels, lymph capillaries, these originate as blind end tubes. They have the same structure as blood capillaries, single layer, their walls are more permeable to protein, cell debris and others. So, the tiny capillaries join up to form large lymph vessels, and the wall of lymph vessels have 3 layers like the blood vessels. The lymph vessels have valves like veins to ensure the lymph flows in one-way system towards the thorax. And the lymph vessels become larger as they join together, eventually forming 2 large ducts, the thoracic duct, the right lymphatic duct. And these 2 ducts empty lymph to subclavian veins. So thoracic duct, it is the largest duct and it begins at the cisterna siltili. The cisterna is a dilated lymph channel situated in front of the bodies of first 2 lumbar vertebrae, right? It extends from the abdomen through diaphragm and runs next to the aorta, and the duct is about 40 cm long and opens into the left subclavian vein into the root of the neck. It drains lymph from both legs, the pelvic and the abdominal cavities, the left thorax, head and neck, and the left arm. Then we have the right lymphatic duct, which is a dilated lymph vessel about 1 cm long. It lies in the root of the neck and opens into the right subclavian vein. It drains lymph from the right half of the thorax, head and neck, and the right arm. How can lymph flow in the lymph vessels? Large lymph vessels has intrinsic to contract rhythmically lymphatic pump. Compression on the lymph vessels due to activity of structures adjacent to these vessels helps to push the lymph along. Structural activity such as skeletal muscle contraction, thoracic pump during respiration exercises accelerate lymph flow. And valves. The lymph nodes. Lymph nodes are encapsulated spherical or kidney-shaped organ composed of lymphoid tissue that are distributed throughout the body along the course of the lymphatic vessels. Now, lymph nodes present everywhere in the body except brain. The lymph drains through a number of nodes, usually 8 to 10 before returning to the venous circulation. 4 or 5 lymphatic vessels enter the lymph nodes. These vessels are called afferent lymph vessels, and only 1 lymph vessel leave it, which is called the efferent lymph vessel. The lymph nodes have a convex side and a concave depression side. It is called the hilum. The hilum, where artery and nerve center and the vein and lymph vessel leave the lymph node. Okay. So both T and B lymphocytes are present in the lymph node, and the lymphatic follicles are composed of B lymphocytes. The diffuse lymphoid tissue intervening between nodules is made up of mainly of T lymphocytes, and T lymphocytes are also present in the medullary cords. The plasma cells derived from the B lymphocytes, macrophages, fibroblasts are also present. So afferent lymphatic vessels pour the lymph into the subcapsular sinus, then lymph passes through intermediate sinuses to reach medullary sinuses, and it's called efferent lymphatic vessel at the hilum. Okay, I will not go into too much detail of this. I will just upload the slide. You can just check through it. These are histological images of the lymph nodes I've taken from a slide. No need to go into too much detail, just for your own sake. So coming back to the question that we started off this first question of this session, which of the following statements about lymph is true? The right answer will be lymph is rich in antigens that originate from tissues. So I will wait for some time. Any doubts you can ask now. Okay, so moving on to the next question for today. Which of the following cells is primarily responsible for antigen phagocytosis in the skin? Option A, M cells. Option B, Langerhans cells. Option C, plasma cells. Option D, epithelial cells. Option E, osteoclast. I want the answer in detail. So anybody would like to try? Okay, so the right answer for this question will be B, option B, Langerhans cells. So, what are Langerhans cells? Langerhans cells are basically specialized antigen presenting cells or APCs found mainly in the epidermis of the skin. So, they belong to the dendritic cell family and play a key role in the skin immune defense system. Their major functions include phagocytosis of antigens like bacteria, viruses, foreign particles, processing antigens, presenting antigen peptides to the T cells. Thus, they are the primary cells responsible for antigen uptake and phagocytosis in the skin. Now, how Langerhans cell work? Step by step, antigen entry, pathogens or foreign substances enter through the skin. Then we see phagocytosis, Langerhans cells capture and engulf the antigen. Then we see antigen processing. The antigen is broken into peptide fragments inside the cell. Then we see migration to the lymph nodes. Langerhans cells migrate through lymphatic vessels to nearby lymph nodes. Then we see antigen presentation. They present the antigen to the T lymphocytes via MHC class II molecules, initiating the adaptive immune response. This process connects innate immunity and adaptive immunity. Why the other options are not correct? Option A, M cells. Microfold cells or M cells are found in the intestinal epithelium, particularly over the Peyer's patches. Their function is basically transporting antigens from the gut lumen to immune cells. They are not present in the skin, so they are not responsible for skin antigen phagocytosis. Option C is plasma cells. They are antibody secreting cells derived from B cells. Their function is producing immunoglobulins or antibodies. They do not perform phagocytosis. Option D is epithelial cells, which is also incorrect because epithelial cells form the protective barrier of the skin. Their function is providing physical protection, producing antimicrobial peptides. However, they do not primarily phagocytose antigens. Then we have option E or osteoclasts. Osteoclasts are multinucleated cells involved in bone resorption. Their function is breaking down bone tissue during remodeling. They are found in bone, not skin, and are unrelated to antigen phagocytosis. So the correct answer will be Langerhans cells, right? So we have talked about the progenitor hematopoietic stem cells and the progenitor cells. I am not going into the details again. We'll directly talk about I'll actually upload these slides so you can just go through this again. We have discussed this a lot. So if you talk about the phagocytic cells, they are monocytes and the derivatives, which are monocytes in blood, dendritic cells in skin, surrounding tissues and lymph nodes, Kupffer cells in the liver, alveolar macrophages in the lung, macroglia cells in the brain, osteoclasts in the bones, synovial A cells in joints, mesangial phagocytes in kidneys, Langerhans cells in skin, macrophages in the peripheral tissues, and option B will be neutrophils. Okay, so what are the steps of phagocytosis? Number one is recognition of the foreign body, then inflammation, then chemotaxis, then adherence, then engulfment, which is endocytosis or pinocytosis, then formation of phagosome, formation of phagolysosome, fusion of phagosome with the lysosome, then killing and digesting of the microorganism, and the disposal and presentation. So recognition. So this can take place by interaction of the pattern recognition receptors (PRRs) and the pathogen associated molecular patterns or PAMPs. Okay. Then the inflammation. Basically, after recognition of the foreign body, the inflammatory process starting by the secretion of the inflammatory mediators by the mast cells and the basophils. So mast cells are resident in tissues near the site of infection. While the basophils are circulating in the blood, the inflammatory mediators cause the following, like increased blood supply to the area of infection, increased vascular permeability due to interaction of the endothelial cells, migration of large molecules to reach the site of infection like C5a, C3a, the migration of the leukocytes in early stage, the neutrophils, and in late stage the macrophages. Then this process is controlled by the C5a, mainly the chemotaxin. Now, chemotaxis of the phagocytic cells can occur by secretion of C5a, C3a, and acute phase proteins. So, this process leads to the migration of the phagocytic cells, the macrophages, and the neutrophils to the site of infection. Adherence. Adherence means the adherence of the microorganism to the cell membrane of the phagocytic cells, which leads to the engulfment of the microorganism by the phagocytic cells. The adherence takes place by one or more of the following opsonizing factors, opsonization like C-reactive protein, C3b, Fc receptors, hydrophobic forces, PRRs, PAMs. Engulfment. In this process, engulfment of the foreign body happens by the bulging of the cell membrane of the phagocytic cells, either by the endocytosis or pinocytosis. In this process, the phagosome is formed, which is a vacuole containing the ingested foreign body. Then fusion of the phagosome occurs with the lysosome to form the phagolysosome. Now, then we talk about the killing, digestion, disposal, and the presentation of part of the foreign body. So, after formation of the phagolysosome, killing and lysis of the foreign body takes place. The killing process takes place by the effect of the oxygen radicals like nitrous oxide and hydrogen peroxide, and the lysis by the lysosomal hydrolytic enzymes. Now, presentation and disposal of the antigens. The final stage of the phagocytosis is the presentation of the antigen to the T lymphocytes by the interaction between the antigen and T cell receptors and MHC molecules. Also, presentation of the antigen to the B cells will occur. Disposal of the undigested part of the foreign body will be later disposed out of the phagocytic cells. We have talked about these. So pathogen infects the human. This is pathogenesis antigen. It mark is an invader. Now, white blood cell or the phagocyte will recognize the pathogen as an invader and engulf it through phagocytosis. The phagocyte will activate helper T cells. The phagocyte now displays the antigen from the pathogen, APCs, antigen presenting cells. The phagocyte helper activates helper T cells. Helper T cells activate B cells which release antibodies. These antibodies attach to the pathogen and signal them for destruction. So now, if we talk about we have talked about antigen, which is defined as an substance that satisfies two distinct immunology properties: immunogenicity and antigenicity. Now, immunogenicity is basically the ability of an antigen to induce immune response in the body, both humoral and or cell mediated. So B cells plus antigen will be effector B cells, plasma cells plus memory B cells. T cells plus antigen will give rise to effector T cells, helper T cell or cytotoxic T cells plus memory T cells. Now, substances that satisfies this property, that is immunogenicity, more appropriately called the immunogen rather than the antigen. Now, what is antigenicity? Which is the immunological reactivity. Now, ability of an antigen to combine specifically with the final products antibodies or the T-cell surface receptors. All molecules having immunogenicity property also show antigenicity, but the reverse is not true. Like haptens, which are antigenic but not immunogenic. Now, epitope or antigenic determinant. Smallest unit of antigenicity. Definition is like small area present on the antigen comprising of few amino acids or monosaccharide residues that is capable of sensitizing T and B cells and reacting with specific site of T cell receptor or an antibody. A specific site of an antibody that reacts with the corresponding epitope of an antigen is called paratope. If we talk about the types of epitope, sequential or linear epitope present as a single linear sequence of few amino acid residues. Now the conformational or non-sequential epitopes found on the flexible region of complex antigens having tertiary structures, formed by bringing together the surface residues from different sides of the peptide chain during its folding into tertiary structure. Now T cells recognize sequential epitopes while B cells bind to the conformational epitopes. If we talk about haptens, low molecular weight molecules that lack immunogenicity, like cannot induce immune responses but retain antigenicity or immunological reactivity, like they can bind to the specific antibody T cell receptor. Haptens can become immunogenic when combined with a larger protein molecule called carrier. The hapten carrier complex is capable of inducing immune response in their body. Now, it is observed that animals immunized with such hapten carrier conjugate produce antibodies specific for epitopes of hapten, unaltered epitopes on the carrier protein, and new epitopes found by the combined parts of both the hapten and the carrier. Haptens contain two or more epitopes. Simple haptens contain only one epitope. Now, based on the antigen-host relationship, antigens can be grouped into two groups: self or autoantigens, non-self or foreign antigens. Belong to the host itself, not immunogenic. Hosts do not react to their own antigens by exhibiting a mechanism called immunological tolerance. Sometimes the self-antigens are biologically altered and can become immunogenic. Now, immunogenic and are three types of based on the phylogenetic distance to the host. Alloantigens are species specific, like tissues of all individuals in a species contain species specific antigens. Isoantigens are type of antigens which are present only in subsets of a species, like blood group antigens and histocompatibility antigens. These histocompatibility antigens are highly specific as they are unique to every individual of a species. Then we have the heteroantigens, antigens belonging to two different species called heteroantigens, like of a plant or animal or microorganisms. Then we have heterophile antigens, which are type of heteroantigens that are present in two different species but they share epitopes with each other. Now, what are the factors that influence immunogenicity? Size of the antigen, chemical nature of the antigen, susceptibility of the antigen to tissue enzymes, structural complexity, and foreignness to the host. Genetic factor, optimal dose of antigen, route of antigen administration, repeated number of doses of antigens, multiple antigens, and effect of prior administration of antibody to the body. Now, size of antigen, larger is the size, more potent is the molecule as an immunogen. The molecules of greater than 10,000 Dalton molecular weight can only induce immune response. Now, proteins are stronger immunogens than carbohydrates, followed by lipids and nucleic acids. Only substances that are susceptible to the action of tissue enzymes are called immunogenic. Degradation of the antigen by the tissue enzymes produces several immunogenic fragments having more number of epitopes exposed. Structural complexity, simple homopolymers made up of single amino acid lack immunogenicity. Polymers made up of two or more amino acids are immunogenic. Addition of aromatic amino acids increases immunogenicity. Foreignness to the host, key factor which determines immunogenicity. Higher is the phylogenetic distance between the antigen and the host, more is the immunogenicity. Now, genetic factor, different individuals of a given species show different types of immune responses towards the same antigen. Responders are the individuals who produce antibody faster. Slow responders are individuals who produce antibodies slowly and may need repeated antigenic exposures. Non-responders are the individuals who do not produce antibody in spite of repeated antigenic exposures. Optimal dose of antigen. An antigen is immunologically active only in the optimal dose range. Immune response is better induced following parenteral administration of an antigen. Site of infection may influence immunogenicity. Repeated doses of antigens over a period of time are needed to generate an adequate immune response. This is due to the role of memory cells in secondary immune response. However, after a certain dosage of antigens, no further increase in antibody response is seen. When two or more antigens are administered simultaneously, the effects may vary. Antibody response to one or the other antigen may be equal or diminished or enhanced. Adjuvant is like any substance that enhances the immunogenicity of an antigen added to vaccines to increase the immunogenicity of the vaccine antigen. Can be alum like aluminum hydroxide or phosphate mineral oil, liquid paraffin fronds, incomplete adjuvant, so water in oil emulsion containing a protein antigen in the aqueous phase. Fronds complete adjuvant is a mixture of fronds, incomplete adjuvant, and suspension of cultivar bacilli in the oil phase. Lipopolysaccharide fraction of the gram-negative bacilli, other bacteria or their products. Okay, we'll not go into too much detail. Started with the Langerhans. I'll upload all of this so that you can go through it. I'll not go into too much detail because we have already discussed so many of this in all the questions that we have been doing. The correct answer will be Langerhans cell. Which of the following cells is primarily responsible for antigen phagocytosis? In the skin, it will be Langerhans cells. Any questions you can ask? Okay, then we'll go to the next question. Uh, what is the one second. What is the significance of the density gradient centrifugation method in immunological research? Option A, it is used to transport blood samples. Option B, it isolates peripheral blood mononuclear cells PBMCs, providing a reliable source of immune cells for experimentation. Option C, it enhances the growth of red blood cells. Option D, it is a method for measuring blood viscosity. And option E, it allows for the direct observation of immune cell function. Anybody? Okay. So the correct answer here will be option B, obviously. Um, it isolates peripheral blood mononuclear cells or PBMCs, providing a valuable source of immune cells for experimentation. So what is density gradient centrifugation? Let's talk about that first. Density gradient centrifugation is the laboratory technique used to separate cells based on the density using a centrifugal force and a density medium, commonly Ficol or Percol. It is widely used in immunology, cell biology, and clinical research. So, how the method works: blood is collected and layered carefully over a density gradient medium, like Ficol. The tube is placed in a centrifuge. During centrifugation, cells separate according to density. The typical layers will be: the top layer is plasma, PBMC layer is the buffy coat, lymphocytes and monocytes, and Ficol layer. Then we have the granulocytes and red blood cells at the bottom. Now, what are PBMCs? Peripheral blood mononuclear cells are immune cells with round nuclei containing T lymphocytes, B lymphocytes, NK cells or natural killer cells, and monocytes. These cells are extremely important for immunological experiments. Now, why PBMC isolation is important in immunology? Density gradient centrifugation allows researchers to obtain pure immune cells for experiments such as immune response studies, cytokine production assays, T cell activation experiments, vaccine studies, flow cytometry assays, cell culture experiments. This provides a valuable source of immune cells for experimentation, making option B correct. Now, why the other options are not correct? Option A is used to transport blood samples. That's not true. This method is not used for transport. It is used after blood collection to separate cells. Why option C is not correct? Enhances the growth of red blood cells. That is not true. Density gradient centrifugation separates cells. It does not simulate cell growth. Option D is not correct because it's not a method for measuring blood viscosity. Blood viscosity is measured using viscometers, not centrifugation techniques. Option E is also incorrect because it does not allow direct observation of immune cell function. This technique isolates immune cells, but functional observation requires additional assays. Okay. So types of centrifugation, if we talk about, it's basically based on the purpose. It can be analytical or preparative. Analytical basically is measuring the physical properties of the sedimenting particles such as sedimentation coefficient or molecular weight. Preparative will be isolating specific particles which can be reused. If we talk about analytical ultracentrifugation applications, basically they can determine the sample purity, characterize assembly and disassembly and mechanisms of biomolecular complexes, detect and characterize macromolecular conformational changes, measure equilibrium constants and thermodynamic parameters for self and heteroassociating systems, and characterize the solution state behavior of macromolecules under various conditions. Now, analytical ultracentrifuge is preparative and ultracentrifuge plus optical detection system. It can measure sample concentration inside the centrifuge cell during or after sedimentation. Now, centrifugation parameters and data acquisition under computer control through experiments lasting many days performed with minimal operated intervention. Okay, so this is the absorbance optical system, measurement of sample concentration at wavelengths from 200 to 800 nanometer, detection of macromolecules containing strong chromophores. Now, if we talk about the Rayleigh interference optical system, measurement of sample concentration based on refractive index changes, analyzing macromolecules lacking intense chromophores like polysaccharides and samples that contain strongly absorbing buffer components like ATP, GTP. Now, preparative centrifugation has many types. Differential centrifugation, self-fractionation. The process of separation of cell organelles is known as self-fractionation. To isolate the specific organelle, the organs are homogenized in a suitable medium at 4 degrees Celsius. The resulting suspension is called homogenate. Now, fractionation is done by differential centrifugation. This method is based upon the differences in the sedimentation rate of particles of different sizes and density by using a series of centrifugation steps at successively greater speeds. Each step can yield a pellet or supernatant. The supernatant from each step is subjected to centrifugation in the next step. It provides 4 pellets: nuclear, mitochondrial, lysosomal, and macrosomal fractions. At each step, the pellet is washed several times with splitting in the homogeneous medium followed by centrifugation under the same conditions. You can see over here. Now, what is density reading centrifugation? Basically, it uses a medium that has gradients like cesium chloride, cesium sulfate, sodium bromide, glycerol, dextran, etc. And separation depends upon the power and densities of the particles. Two types: rate-zonal and isopycnic. Now, rate-zonal centrifugation is gradient used as a maximum density at the bottom, but density is less than that than the most dense sedimenting particle to be separated. Density gradient is shallow. Sample is taken at the top as a zone. Centrifugation is performed at low speed for a short time. Now, depending on the sedimentation rate, samples from discrete zones must be determined before the zones reaching the bottom. Okay. An application is useful for separating proteins with nearly identical densities but differing slightly in size. It's used for separation of DNA RNA hybrids, ribosomal subunits, and subcellular organelles. When we talk about isopycnic centrifugation, it depends on the power and density of the particle. It does not depend on shape or size of the particle, independent of time. Maximum density of the gradient always exceeds the density of the densest particle. At the point of isodensity, no further sedimentation occurs. Okay. The application can be used to separate particles of similar size but of different density. Subcellular organelles such as Golgi apparatus, mitochondria can be effectively separated by this method. Now, centrifugation is the technique of separating components where the centrifugal force or acceleration causes the denser molecules to move towards the periphery while the less dense particles move to the center. The process of centrifugation relies on the perpendicular force created when a sample is rotated about a fixed point. The rate of centrifugation is dependent on the size and density of the particles present in the solution, and centrifugation is a technique of separating substances which involves the application of centrifugal force. The particles are separated from a solution according to their size, shape, density, viscosity, medium, and rotor speed. In a solution, particles whose density is higher than that of the solvent sink sediment, and particles that are higher than it close to the top. The greater the difference in density, the faster they move. If there is no difference in density like isopycnic conditions, the particles stay steady. A centrifuge is a piece of equipment that puts an object in a rotation around a fixed axis, like spins it in a circle, applying a potentially strong perpendicular to the axis of the spin outward. Centrifuge works using the sedimentation principle where the centripetal acceleration causes denser substances and particles to move outward in the radial direction. At the same time, objects that are less denser displaced and move to the center. So, relative centrifugal force is a measure of the strength of rotors of different types and sizes. This is the force exerted on the contents of the rotor as a result of the rotation. It is a perpendicular force acting on the sample that is always relative to the gravity of the earth. The RCF of the different centrifuge can be used for the comparison of rotors, allowing the selection of the best centrifuge for a particular function. Centrifuge is a device used to separate components of a mixture on the basis of their size, density, viscosity, and rotor speed. Centrifuge is commonly used in laboratories for the separation of biological molecules from a crude extract. In the centrifuge, the sample is kept in a rotor that is rotated about a fixed point or axis, resulting in strong force perpendicular to the axis. There are different types of centrifuge used for the separation of different molecules, but they all work on this principle of sedimentation. So, for example, if you talk about rotors, centrifuge rotor is designed to generate rotation speed that can bring about the separation of components in the sample. So there are three main types of rotors used in centrifuge, which are fixed angle rotors. So these rotors hold the sample tubes at an angle of 45 degree in relation to the axis of the rotor. And in this type of rotor, the particles check the opposite side of the tube where the particles finally slide down and are collected at the bottom. These are faster than other types of rotors as the path length of the tubes increases. And however, as the direction of the force is different from the position of the tube, some particles might remain at the side of the tubes. These are fixed angle rotors. Then we have swinging bucket or horizontal rotors. Swinging bucket rotors hold the tubes at an angle of 90 degrees as the rotor swings as the process is started. In this rotor, the tubes are suspended in the racks to allow the tubes to be moved enough to acquire the horizontal position. And in this type of rotor, the particles are present along the direction of the path of the force that allows the particles to be moved away from the rotor towards the bottom of the tubes. Because the tubes remain horizontal, the supernatant remains as a flat surface, allowing the deposited particles to be separated from the supernatant. This is a swinging bucket rotor. Then we have vertical rotors. Basically, they provide the shortest path length, fastest runtime, and the highest resolution of all the rotors. In vertical rotors, the tubes are vertical during the operation of the centrifuge. The yield of the rotor is not as ideal as the position of the tube does not align with the direction of the centrifugal force. As a result, instead of settling down, particles tend to spread towards the outer wall of the tubes, and these are commonly used in isotechnic and tensile gradient centrifugation. So benchtop centrifuge is a compact centrifuge that is commonly used in clinical and research labs. So it's driven by an electric motor while the tubes are rotated about a fixed axis, resulting in force perpendicular to the tubes. And because these are very compact, they're useful in smaller labs with smaller spaces. So different variations of the benchtop centrifuges are available in the market for various purposes, and a benchtop centrifuge has a rotor with racked sample tube and a lid that closes the working unit of the centrifuge. Low-speed centrifuges are the traditional centrifuges that are commonly used in labs for the routine separation of particles. So they operate at a maximum speed of 4000 to 5000 rpm. These are usually operated under room temperature as they are not provided with a system for the speed or the temperature of the operation, and two types of rotors are mostly used. Fixed angle or swinging buckets is used for sedimentation of red blood cells until the particles are slightly packed into a pellet and supernatant is separated by decantation. These are easy and compact centrifuges that are ideal for the analysis of blood samples and other biological samples. Then we have high-speed centrifuge, as the name suggests. It is a centrifuge that can be operated at somewhat larger speeds, so they can range from 15,000 to 20,000 rpm. High-speed centrifuge is commonly used in more sophisticated labs with the biochemical application and requires a high speed of operations, and they are generally provided with a system for controlling the speed and temperature of the process, which is necessary for the analysis of sensitive biological molecules. And high-speed centrifuges are used in the more sophisticated biochemical applications. Higher speeds and temperature control of rotor chamber are essential. So the operator of the instrument can carefully control speed and temperature, which is required for sensitive biological samples. So all three types of rotors are available: fixed angle, swinging bucket, vertical rotors. Then we have microcentrifuge for centrifuges of volumes like 0.5 to 2 microliter. They are usually operated at about speeds of very less. This is used for molecular separation of cell organelles like nuclei, DNA, internal extraction. And they are also termed as microfuge, whose sample tubes that are smaller in size when compared to the standard test tubes used in larger centrifuges. And they always sometimes come with adapters that facilitate the use of large tubes along with smaller tubes. So microfuges, basically tabletop spin tubes, are with temperature controls that are available for the operation of temperature sensitive samples. Then we have refrigerated ones, which are provided with temperature control ranging from -20 to -30 and all of that. A different variation of centrifuges available. There is a system of temperature control is essential for various purposes requiring lower temperatures. There were temperature control unit in addition to the rotors and racks sample tubes, and this can provide RCM of 60,000 to 1 lakh, that is ideal for separation of various biological particles. And this is typically used for collecting substances that separate rapidly, like yeast cells, chloroplasts, erythrocytes. And the chamber is refrigerated, centrifuge sealed off from the outside to meet the conditions of operations. Then we have ultracentrifuges. It's a more sophisticated instrument that operates at extremely high speeds that allow the separation of some smaller molecules like ribosomes, proteins, and viruses. It's a more sophisticated type of centrifuge that allows separation of molecules that cannot be separated by other centrifuges. The refrigeration systems are present in such centrifuges that help to balance the heat produced due to the intense spinning. The speed of the centrifuges can reach as high as 1 lakh rpm with maximum speed of 1 lakh g, so it can be used for both preparative and analytical works. They can separate molecules in large bulk batches and in continuous flow systems. In addition to separation, ultracentrifuges can also be used for the determination of properties of macromolecules like size, shape, density, and all of that. So we'll talk about only density gradient centrifugation. This type of centrifugation is mainly used to purify viruses, ribosomes, membranes, etc. The sucrose density gradient is created by gently overlaying lower concentrations of sucrose on higher concentrations in centrifuge tubes. The particles of incrust are placed on top of the gradient and in centrifuge in ultracentrifuges, and the particles travel through the gradient until they reach a point at which their density matches the density of surrounding sucrose. The fraction is removed and analyzed. We will not go into too much detail. Okay, so in this when the substances are concentrated in a solution of calcium, cesium salts or sucrose, and the method involves the fractionation of particles based on the Bowen density. Okay, Bowen is basically the ability or the tendency of something to float in water or other fluid to keep something afloat. So density gradient in this method is used cesium salt or the sucrose medium. Okay, so it's based on the principle that molecules settle down under the centrifugal force until they reach a medium with the density the same as theirs. So in this case, a medium with the density gradient is employed, which either has to decrease density or increase in density. When the molecules in the sample move through the medium as the sample rotates, creating a centrifugal force. The more dense molecules begin to move towards the bottom as they move through the density gradient. The molecules then become suspended at a point in which the density of the particles equals the surrounding system medium. In this way, molecules with different densities are separated at different layers, which can then be recovered by various processes. So if you see over there, this can be lysosomes, mitochondria, peroxisomes will go at the bottom. So a density gradient is created by gently laying the lower concentration over the higher concentrations in a centrifuge tube. The sample is then placed over the gradient and the tubes are placed in the ultracentrifuge. The particles travel through the gradient until they reach a point at which the density matches the density of the surrounding medium. The fractions are removed, separated, obtaining the particles as isolated units. In rate-zone centrifugation, the sample is layered as an arrow zone on top of the density gradient. Particles move at different rates under the centrifugal force on the basis of the density. The actual sedimentation rate primarily depends upon the size and mass of the particles. As the density of the particles is higher than the density gradient, all particles produce pellets. Okay, if you see from here, isotechnic centrifugation is the second type of density gradient centrifugation method. It starts with a homogeneous solution. Under the centrifugal force, the particles in the analyte mixture move until the density of the particles is similar to the density gradient. Therefore, we can name this technique as equilibrium centrifugation as well. So it can be applied for purification of large volumes of biomolecules. It can be used for purification of different viruses, which aids their further studies, and can be used as both separation technique and technique for the determination of densities of various particles. So this is differential and density gradient centrifuge. So this technique was used by mesenchymal star also, that utilizes the cesium chloride as a medium. A gradient of densities from lowest to highest is formed and components remain in the layer that corresponds to the densities. So it's basically a cesium chloride solution is set up in the centrifuge tube and it forms a concentration gradient within the tube when centrifuge at high speed with more concentrated cesium chloride towards the base. Because cesium is a heavy element, the cesium salt solution is much denser than the density of most salt solutions. The cesium salt do not affect viruses or DNA. It is used in centrifugation. Okay. So basically, coming back to the question that we started off, uh, density gradient centrifugation is used to isolate peripheral blood mononuclear cells or PBMCs, providing a reliable source of immune cells for experimentation. That is the answer to this question. Option B. Any doubts? Any questions? So, if nobody has any question, we can stop the session for today and we'll meet again next Monday, same time. Thank you. Okay, if nobody has any question, we'll stop the session.